we mentioned with you that the fundamental structural unit of the matter is atom. Of course, atom consists of the smaller particles in it, as you can see on this picture: proton, neutron, electron. The atom itself consists of the two main parts, the nucleus and electron. Okay, I'm sure this is simple things that you already know, but we need to go through them because maybe somebody needs a refreshment. Because the as you can see in the atom here, you have the electron that is spinning around the nucleus, and it has negative charge. The nucleus here is made up from protons and neutrons. So the protons, they carry positive charge that opposes the negative charge of the electrons. And so depending on the numbers of the protons and electrons, you will have the charge, what's called the electric charge of this, in that case, ion. If it has a charge, it will be called ion. But in the normal state, all these elements, they are neutral because they will have equal number of electrons and protons. And the neutron is not charged because neutron itself is made from proton and electron. Okay, so that's why neutron is neutral, and it only adds to the mass of the element, of the atom. So we have the atomic number is the number of protons in the nucleus, and then atomic mass is the number of protons and neutrons combined. Okay. Then also fundamental understanding or idea about the different elements, atoms, is the energy levels of the electrons that are spinning around them. The further electron, like here in this picture, the further the electron away from the nucleus, the more energy it has. The closer it is to the nucleus, the lower level of energy it has. And these levels of energy are called levels of shells, energy levels or shells. And for the most part, we will be dealing with the, and also each of these levels has a certain amount of electrons that it can accommodate, okay? So it can only accommodate certain amount of electrons. For example, the very first one can accommodate only 2 electrons. So that's the very first level. And then the second one can already accommodate 8 electrons. And they come in a different formation. So that means they're spinning around the nucleus in a different, they create different forms, which we're not going to go around. We're not going to talk too much about it because this is really not required for this course. But what we need to mention is that the most important for us, the most important shell in the atom is the outermost shell. Why is that so? Well, that's because the outermost shell is where the reactions takes place. These electrons will interact with the electrons from another atom, and they can form different bonds, what is called. So the outermost electrons, they're called valence electrons, which we're going to see that word in the future. We're going to talk more. I probably will just write here. It's called valence electrons. These are the valence electrons. These are on the outermost shell. And so they will define how this atom will behave in relation to other atoms. All right. So next is the difference between elements and compounds. Elements are always made up from the same atom. only one atom, let's say pure gold, right? Or pure oxygen. So that would be an element. Even though we know the difference between atom is only one. Atom is different from molecule. Molecule is made up from two or more atoms, as you can see here. So here's the molecule O2, or H2O is made up from more than one atom. And it can be the same atom or same element or from different elements. So if it is the same element, and as you can see here, oxygen has the O2 form, that means you have two atoms of oxygen connected to each other, and they're sharing electrons with each other. So this is still going to be element, even though there are more than one atom that makes the molecule here, it's still called element. Compound made up from more than one element. As you can see, water. Water is made from hydrogen and oxygen, right? So hydrogen and oxygen, two different elements, make up a compound. All right. Okay, I already said about the bonding, and we will look with you how those bonds actually created, because there is a certain principle, but it's better to explain on this picture to you. So, as I mentioned earlier, that different atoms, they have different number of electrons that are rotating around on these orbits. And every shell or every level, as we mentioned, can accommodate only certain number of electrons. And interestingly enough, that the different atoms, they're trying to get to the condition where the most stable. And they're the most stable when the number of those electrons in the outermost shell, in this case, the outermost shell, is as close as possible to the number of electrons that it can contain. Okay, so for example, we will, as we look with you at this picture, let's count. This is salt, sodium chloride. So sodium has only one electron on the outermost level, right? You see that? Only one electron. And now let's look at the chloride. How many chloride has? 1, 2, 3, 4, 5, 6, 7. And what's interesting that this level is comfortably can accommodate 8 electrons. So chlorine is very hungry right now for that extra electron. And interestingly enough, sodium is actually ready to get rid of it because it will feel more comfortable without anything in the outermost shell. And so what will happen is that the sodium will donate electron, as you can see, that's what happened here. It will donate its electron to the chloride, and what is going to be formed? The crystal salt. Okay, when the salt crystallizes, this is what you see. You see the crystals of sodium chloride. Of course, if it is in the water, then this ionic bond will be broken down, and they're not going to be next to each other like this, okay, because the water will be separating them. But once it crystallizes, then this ionic bond will be created, and they will be together. They will be together. Otherwise, it will simply donate the electron, but it's not going to stick together because ionic bond is not very strong. It's actually not very strong, and once you put it in the water will break down this bond.
Quick question. So these only happen when it is a compound, you said?
Okay, so no, actually, this sharing of electrons can take place between elements and between the same kind of elements and different elements. For example, let me see. Here's another example. So you have two hydrogen atoms, right? Both of them, so they're similar. It's not like with this example,
Right.
you have sodium chloride. They are the same. And each one of them has only one electron in the outermost shell, right? But this is the very first level. So remember, it's the first, hydrogen is the most simple chemical, is the very first chemical in the table of the chemical elements. And so it has one electron, but guess what? On this first shell, it can comfortably accommodate two electrons. So it actually wants to add one more electron if it can. And this is what happens. These two hydrogen atoms, they come together and they form what's called covalent bond. Now, the covalent bond is different from the ionic bond, as that we saw with you a little earlier, is that ionic bond is basically a robbery. That's when one atom will actually steal the electron from another one, okay? Because he is not sharing anything. He just took it. Whereas with covalent bond, it's much stronger bond. And here it's a fair share. It's like a Christian church, early church. Everything was shared, right? In book of Acts, we read that. So, and this is a very strong, actually, bond. It's not like ionic bond. So, and here you can see example where two of the elements, the same elements, they form the bond. It's called, and now we have the hydrogen molecule here. Here's an example I have of hydrogen peroxide. You can see that it's a compound because it's made from two different elements. So you have the covalent bond. It's a double bond here between the oxygen, and then you have what's called hydroxyl group here, where it's actually also a covalent bond, but in this case, I don't really to go too much into details of the hydroxyl white because it's a very powerful free radical. It's a powerful free radical, hydrogen peroxide. So the reason why hydrogen peroxide is very powerful disinfectant is because if we will look, I guess we will look into it. The oxygen can actually take well, it's actually six atoms, six electrons in the outer shell. And so it's hungry for two more. So here in this case, well, I guess I just have to draw to explain what's happening in more details. So here's the oxygen, right? So this is the outer shell. And the outer shell actually is made from two levels. One, it's called s orbital. They call them orbitals. So this is s orbital. And then you have another orbital. Now, this is all on the same level. It's called p orbital, and it has four. Actually, I need to draw symmetrically. Just a minute, let me erase this. Okay, so one, sorry, I need to draw another circle here. And so you have one here, one here, one here, and one here, four. Okay, so it actually can take six, and it's called p orbital. It has a different shape. It actually has this kind of shape, whereas this s orbital has this shape. It's round. It's like a cloud, like a ball. So now these four electrons are the ones that will want to combine with whatever. And so what we have here is that, you see, it has altogether 6. So it's missing 2 more. So as you can see, you have a double bond here. So 2 of them already combined. Then you have 1 here. Actually, sorry, not here, these ones. Let me erase again, just to make it a little bit clear. Give me just a second, I'll redraw it. And so here is 1. So these ones on the outside, so 2 of them already connected. See, because you have double bond here between 2 oxygen atoms. Then there is 1 more already also occupied, this one. Okay, so this one is occupied. Now it has 1 more missing. It has 1 more electron that is ready to connect with something, and it does not have the extra electron. So that's why the hydrogen peroxide will react with whatever, because it has a missing pair. It has 1 electron that is not paired yet, and it's looking for that extra electron from whatever it's going to get in touch with. Maybe it could be bacterial membrane, or maybe whatever it is. And so that's why in itself it's neutral. There is no charge here. Okay, it's neutral. That's the difference between ions and free radicals. Free radicals, they don't have a charge, but they are hungry because they want to pair their electron. Okay, they want to pair the electron. And this one is even more powerful because you have 2 oxygens that are very hungry, and they're looking for another atom, another element, whatever it is, to get in touch and to take from them, to oxidize them, to take from them that electron. All right. Let's get back. And so in this case with ionic bond, you have the now 2 ions. Now they're charged. Why they're charged? Because this sodium lost 1 electron, so now it is positively charged because it has more protons now than electrons. And the chlorine has more now electrons than protons, so it has negative charge. And of course, they will attract each other because the opposite charges will attract. Okay, so that's ionic bond. Right, we already talked about the covalent bond. That covalent bond, they do not ordinarily easily dissociate in water. They are very strong. You have to really heat up the molecule. You have to really heat it up to dissociate those bonds. And then finally, we have what's called hydrogen bonds. Hydrogen bonds, they're the weakest ones. They are more like attractions between the different parts of the molecule. So let's say you have 2 water molecules, right? H2O. Here's 1 water molecule, here's another water molecule, and they form what's called hydrogen bonds. Why hydrogen bonds? Is because, so as we're looking here at this picture, you have oxygen and 2 hydrogens, and even though it's a covalent bond, but the hydrogen is, the oxygen pulls hydrogen closer to itself, and it creates this uneven charge, because you would think that this hydrogen, they should be kind of on the same line away from each other, but this uneven position of the bond between oxygen and hydrogen creates this kind of angle. So they are on the angle, which creates, again, in turn, polarity of the molecule, because hydrogens, they are will have more positive charge, and oxygen will have more negative charge because it's pulled to itself the electrons, right? So let's say if we will look here, so let's say I'll just draw here oxygen, and here's hydrogen. So you would think that the electron should be like exactly in the middle, but in this case, it's not the case. And the electrons, they're closer to the oxygen. And that's why you have this uneven charge, because now oxygen become negatively charged and hydrogen positively charged. Okay? Like here, for example, see, they're exactly in the middle, and it's a neutral molecule. It's a neutral molecule because the electrons are exactly in the middle. They equally share them, and so none of them gets more charge than another. And because of this difference in charges now, we have polar molecule, what's called polar molecule. Now, this is really important idea in chemistry, polarity of the molecule, because you have polar and non-polar molecule. So you have molecules that have no charge, and you have molecules that have charge. So example of the non-polar molecules would be, for example, oils, fats. They are non-polar. An example of carbon dioxide is also non-polar. Okay, and this polar and non-polar molecules, they do not mix with each other. Okay, they actually do not mix with each other. Polar substances, they mix with each other, and non-polar substances also mix with each other. Like you can mix different oils, but you cannot mix water with the oil, right? Okay, well, we have a break as we are talking about the hydrogen bonds. So as we mentioned, the polar substances, they mix only with the polar substances. Non-polar substances, they mix only with non-polar substances. And that plays actually important role, as we will see in the future with you, in the life of the cell. It's a very important mechanism. For example, that will define the permeability of your cell membrane, that not every substance can just go through it. Why? Because of the same principle that we just described with you. There are polar substances, like the water, for example, and non-polar, like oils, fats. They are non-polar. They don't have any charge. That means there is no any kind of atom there that would steal from somewhere and got an extra charge. So it's all neutral, basically. That's what it is. It's all neutral. Whereas the polar substances, they have uneven distribution of electrical charge in them, you know, that comes between the electron and proton interplay. All right? And so this hydrogen bonds is what holds the water together, right? That's what holds the water together. And as you familiar very well with the water, water is very fluid. That means these bonds, they can easily break and be rearranged. So that's why another word for this hydrogen bonds is more like an attraction. So it's not a real bond that is really strong. It's not a strong one. It's fairly weak. Okay. So the chemistry is divided into organic and inorganic, of course, chemistry. So there are organic substances and inorganic substances. So how do they differentiate between them? Well, in general, the organic molecules, they will contain carbon-carbon covalent bond. So you know the word covalent now. That covalent means that they share equally the electrons that join them together. And it's a strong bond, remember that. So they have carbon-carbon or carbon-hydrogen bonds. Here's the carbon-hydrogen bonds. Okay, so either this type of bond or this type of bond. Okay, so the example would be alcohol. This is the alcohol, C2H5OH. There's the alcohol. please notice here that it has this hydroxyl group that we just mentioned with you. And just in case you was wondering why we use the alcohol to clean our hands, it's because of this hydroxyl group, because it's hungry for an electron. And so it will, when it comes in touch with bacteria on our hands, it will destabilize them and often inactivates different pathogens. This is what alcohol does. So this is ethyl alcohol, ethyl alcohol. So what would be examples of the inorganic molecules? Water, acids, bases, and salts. So let's look with you at the chemical reaction that takes place between, in this case, organic molecules. So what we're dealing here is we're dealing with, on this example, with, for example, sugars. Okay, sugars. So sugars are organic substances. They have plenty of carbon, and carbon-hydrogen bonds. For example, it can be glucose joining glucose or glucose joining fructose, you know, the simple sugars that form disaccharides. And of course, you have simple sugars, what's called monosaccharides. Let me see if I can show you the slide with mono. Here we go. Monosaccharide. Then you have disaccharides and polysaccharides. So this is just an example we're going to use to discuss the chemical reaction between these organic molecules. So this is the formula of the glucose. Okay? And examples of the monosaccharides would be glucose, fructose, and galactose, okay, or milk sugar. Then you have disaccharides, and disaccharides made from two monosaccharides, for example, glucose and fructose, will make sucrose, and the sucrose is the table sugar that we are so accustomed to. Or, for example, two glucose molecules can join together and form maltose. That's the barley malt that we know, or maybe the barley malt that we know. And lactose is the milk sugar, is a combination of glucose and galactose. Okay, so now let's look how they combine together. So here we have what's called synthesis, dehydration synthesis. So you have this one organic molecule that joins with another organic molecule. So here's one organic molecule and here's another organic molecule. And as you can see, the way they join together, the way that those, let's say, glucose combines with another glucose molecule, for example, to form glycogen in the liver, that's the reaction that takes place all the time, especially after the meal, as the liver replenishes the glycogen storage. And glycogen is similar to starch in plants. That's the source of fuel for your body. Okay, so let's see how liver accomplishes it. Well, of course, with the help of enzymes, but the bottom line that this glucose will join this glucose by losing, one of them will lose the hydroxyl group and another will lose the hydrogen group. And so hydrogen will join the, in this case, hydroxide, because once it leaves the molecule, it already will be called hydroxide, and they will form water, H2O, right? So this is what's called dehydration synthesis, because they lost a water molecule combining with each other. So two glucose molecule, now they formed a disaccharide that is called the dextrose. You know, you actually, maltose. maltose. Well, dextrose is another name for the glucose, but the maltose is the combination of two glucose molecules. Okay, two glucose molecules, when they join together, they form disaccharide that is called maltose. Now let's look, for example, now let's say I did my exercise, so my sugar level begin to drop, my glucose level begins to drop in the blood. So what the liver is going to do is going to break down the maltose that's basically forms the glycogen molecule, because glycogen molecule is a polymer. It's a polysaccharide. So it has many of these disaccharides now together, right? And so to break it down, it has to actually bring water molecule to dissociate two glucose molecules apart, you know, to take them apart. And now one of the glucose molecules will take the hydrogen part and another will take the hydroxide, and it will become a hydroxyl group in another one. And so basically, so that's what happens. Dehydration synthesis, you lose the water, and with the hydrolysis, you actually, with the dehydration synthesis, you produce water. And with the hydrolysis, you actually lose water. You lose water. And that's interesting. For example, if you ate a lot of sweets, you know, you get thirsty, right? When we eat a lot of sweets, one of the reasons that we get thirsty after eating a lot of sugar is that because for the body to break down carbohydrates, whatever it is, potato, or if it was an apple juice, you know, where you have either disaccharides or polysaccharides, the complex carbohydrates, still your body will have to use, with the help of enzyme, this process where it will be using this water as a knife. So the water is like, you can imagine, as a knife. So the word hydrolysis means, hydro means water, and lysis means to break apart, okay? To break. Lysis, to separate, to break. So breaking water, breaking with water, you may say, breaking water. So, and that's what exactly happens. And so the body is using up the water when it's trying to metabolize your complex carbohydrates, all the way down to the disaccharides. Okay, so to break down those bonds, it has to, it has to use water. Next is, let's talk about acids, bases, and salts. Of course, we know that the water is neutral, even though in the liquid state, of course, water, it dissociates into hydrogen ion and hydroxide ion. And hydroxide ion is negatively charged, and hydrogen ion is positively. Now, the reason why this hydroxide ion is formed in the first place with the negative charge is because it took electron from hydrogen. So now hydrogen lost its electron, but the hydroxide actually gained an electron. And the reason why it's gained, as you just remember not long ago, I was describing you that oxygen is desperate to fill in its outermost shell. And it had only 6, so it can take 8. So it took 1 extra electron, and it's actually doesn't mind to get another electron if it gets a chance. That would increase the charge, of course. All right, so, but they neutralize each other, and that's why water is neutral. So let's see the acid. What makes acid acid is the presence of unbalanced hydrogen ions. So you see in water, it's balanced, right? So that's why water is neutral. But acid has more hydrogen ions than hydroxide ions. And that gives acid the properties that are pertinent to the acids. So because this hydrogen ion is the number of the hydrogen ions will define the strength of the acid. The more of the dissociated hydrogen ions you have, the more is the stronger is the acid. When it comes to base, is the opposite. You look at the number of the hydroxide ions. And so the more of the hydroxide ions you have, the stronger is the base. The more soapy the substance will feel. Okay? So that's the alkaline. So remember, base and alkaline is the same thing. Okay? We say it's the alkaline solution, for example, or it's an alkaline substance, or you can say simply it's base. Will mean the same thing. Okay, so pH is the way that concentration of the hydrogen ions is calculated or evaluated. When the pH is 7, it's a neutral solution. It's neither acid nor base. So if we look here, you see that the 7 is in the middle from 0 to 14, and that defines the neutrality of the solution. It's neither acid nor base. That means you have same number of hydrogen as hydroxide ions, so they balance each other. Now, as you can see that if you have more hydroxide ions, then the solution becomes more basic. If you have more hydrogen ions, it becomes more acidic. And you can look, for example, that your stomach produces pretty strong acid. It's called hydrogen chloride, hydrochloric acid. It's fairly strong acid, 0.8. Look at the orange juice. It's also fairly acidic, 2.8, and so forth. Our urine is slightly acidic. Do you think it's good or bad for our urine to be slightly acidic? Well, it's actually good, because if the urine wouldn't be acidic, then it would be more inviting for our urinary tract would be more inviting for the pathogens, for bacteria, because this mildly acid environment controls and prevents the overgrowth of bacteria in our urinary tract. So if somebody has urine that, let's say, more neutral or basic, then they are more vulnerable and to get UTI, urinary tract infection. All right. So pH is a very important mathematical expression of the concentration of hydrogen ions in the solution. Okay? And of course, if you mix acids with bases, they will neutralize each other, and especially in equal molar numbers, with equal numbers of more acidic substance with the alkaline substance, so they will neutralize each other. For example, in the body, our body is using several ways to maintain that specific pH in the blood. And as you remember, our blood is slightly what? It's slightly alkaline, right? Slightly alkaline. As you can see here on this picture, here's the blood. It shows here 7.4, which is the range between 735 and 745. So 7.4 is about in the middle exactly. All right. Our body will actually do everything possible to maintain this pH, because this pH is important for our life. It's critically important, this range of the pH, for the chemical reactions to take place. And so the body will use several mechanisms. The most effective way of maintaining this pH balance in the blood is our breathing, is the respiratory system. Now, I'm not going to go into all the details how the respiratory system is maintaining it, because we will have respiratory system to talk about it. I will just simply point to you, without much explanation, that carbon dioxide is carried in the blood in the form of carbonic acid. So this is carbonic acid. And this is actually an amazing feat that our body accomplishes by carrying the gases in the liquid form. Okay, that's actually amazing. And so this carbonic acid is a fairly weak acid. Once it reaches the lungs, it decomposes into, actually here, into carbon dioxide and water. And so we lose the carbon dioxide, we exhale it. And then the opposite process take place in the tissues, where it actually picks up carbon dioxide and binds it into the carbonic acid to carry it away to the lungs.
Quick question. An example of a buffer, are these all? These are all buffers, basically, right? The oxygen and all these things that you're saying, the hemoglobin, all these are buffers?
No, actually, no. Wait a minute, I'm going to get to it. Yes, these are the buffer systems that, let me get to it, otherwise I'll lose my train of thought. So right now I'm looking at the mechanism that the respiratory system is maintaining the pH in the blood, maintaining the concentration of the hydrogen ions. So what I wanted to bring your attention here right now is that if, for example, concentration of carbon dioxide goes up in the blood, so let's say I stop breathing, right? So the concentration of carbon dioxide goes up, then this whole equation will shift to the left. Right now, I just simply want to look with you at this equation so that by looking at it, you could tell quickly what is the condition in the body and where the shift is going to take place. Right now, I'm not even talking about how hemoglobin is binding the carbon dioxide or oxygen, because hemoglobin is the buffer, actually, molecule in the cell, in the red cell. And it actually comprises about 75% of the buffering system is just hemoglobin, okay? So, and what I wanted just for you to point so that you would get a kind of idea of how this shifting takes place is when, for example, carbon dioxide begins to build up in the blood, is that this carbonic acid begins to dissociate into hydrogen ion and bicarbonate ion. So this is bicarbonate and this is hydrogen, okay? So as you can see that our blood will right away become more acidic if the carbon dioxide will begin to build up, because if the carbon dioxide begins to build up, then this whole equation will shift to the left, and it will begin to try to bind the carbon dioxide into the bicarbonate ions, and with it you will be getting hydrogen ions. And now you need to do something with these hydrogen ions, and that's where the hemoglobin comes into play, and it binds the hydrogen ions in the red blood cells. So this is just an explanation why when we hold our breath, we can't just do it indefinitely. And the reason, one of the major reasons that forces us to breathe is that the acidity will start going up, because you will start producing too much of the hydrogen ions eventually, and not all of them will be able to be buffered by the hemoglobin. So the next is, let's say I'm hyperventilating, I'm breathing out quickly carbon dioxide. So what then is going to happen? Well, then actually the body will be, then the equation will actually shift to the right, and the more and more of the hydrogen ions will be bind into the carbonic acid, and then the equation will shift to the right, and that's how we will become more alkaline. So basically, simply speaking, by looking at this formula, the easiest way to say is just by looking at this hydrogen ions. What's happening? Let's say if the equation shifts to the left, you're producing a lot of hydrogen ions and you become more acidic. If the equation shifts to the right, then you are binding more and more of these hydrogen ions into the carbonic acid, and with the production of the water and carbon dioxide that you are exhaling, and so you will become more alkaline. So that's why hyperventilation actually makes us more alkaline. When we're holding the breath, we become more acidic. Okay, so, and there are also other buffer systems. For example, kidneys is the second one, because kidneys, as we will look with you also in the future, able actually to retain bicarbonates and to secrete hydrogen ions. That's why urine is slightly acidic, because of the mechanism built in that urine is able to actually secrete the hydrogen ions and retain bicarbonates. So kidneys actually is a reservoir for these bicarbonate ions that actually neutralize the acid. And the third mechanism is actually minerals that can be drawn from the bones. Like, for example, the most common one is the calcium bicarbonate. Here, of course, has calcium phosphate, also calcium bicarbonate, different salts that make up our bones. And so the calcium bicarbonate will actually dissociate into calcium and bicarbonate. And then this bicarbonate group will be used for neutralization of the hydrogen, because bicarbonate will join hydrogen and form this carbonic acid, which is a weak acid. A weak acid. What does weak acid mean? Weak acid means that the strong acid, like for example hydrochloric acid, has very high level of dissociation of the hydrogen from the chloride. With the carbonic acid, you don't have a lot of dissociation. You have a very mild dissociation of hydrogen from bicarbonate. That means, for the most part, in your blood, carbonic acid stays as the carbonic acid. It does not separate into hydrogen ion, which is very aggressive, and then bicarbonate. You see, that's the reason why we have carbonic acid and not sulfuric acid or hydrochloric acid, whatever it is, those strong acids, right? Then we would be really struggling with acidity in our blood. Okay, and so the calcium bicarbonate, as it pulled from the bones, will be used by the body in the long run. I mean, it's not going to do it if I just held my breath for a few minutes, right? But in the long run, if the body detecting that there is a long-standing issue with the body struggling with the maintaining the proper pH, then in the long run, it will start pulling the calcium bicarbonate from the bones. Okay. And which can lead to osteoporosis, of course. Okay.
I had a
So,
question about the saliva. Is that also is that a alkaline or is that
yeah,
acid?
so the saliva is actually alkaline. So actually, yeah, the saliva is saliva is alkaline. Then in your stomach you have the acidity, and that's actually an important. There is a reason why your saliva is alkaline. It's alkaline because when the base mixes with the acid, it actually produces energy, it produces heat. So, so this mixing of the saliva with the acid in the stomach actually raises the temperature. It's called exothermic reaction. Exothermic means it's producing energy. Okay.
What would be the levels of pH for saliva? It's not. I don't see it here.
It's very mild. It's very mildly alkaline. Very mildly alkaline. I don't know exact number, but it's very mild.
Okay.
So we talked already with you about the monosaccharides, the disaccharides, the polysaccharides, and the glucose. That is it's a 6-carbon unit, and 1, 2, 3, 4, 5, and this is the 6th one is right here, the 1 oxygen here. All right. Next is the lipids. Lipids is like an generic. It's like a general term for fats and oils, and the most common form of fat in the body is triglycerides. Triglycerides is the form of storage of the fats in our body, in the fat cells, for example, and in the muscle cells. So in the liver, in the hepatocytes, the fats are bound into triglycerides. So what is triglyceride? Let's look with you at the triglycerides. Here's probably will be a better picture for you. Here's the triglyceride. So the word tri means that there is a three of something, right? And then you have glycer. There's the glycerol. Glycerol is a backbone of the triglyceride. So as you can see, it consists of 3 carbons and 3 oxygens, and then you have hydrogens around it. So this is the glycerol, and this is the tail. The tail is made of the fatty acids. So you have 3 fatty acids joined to the glycerol, and you have your triglyceride. Okay, so this is the glycerol by itself, and as it joins the 3 fatty acids, so it loses 3. So how would you call this reaction based on what you already know? Dehydration synthesis, right? So here's another example. So this can take place with different organic molecules, not only with the sugars that we looked with you a little earlier, carbohydrates, but here we see the fatty acids, they join the glycerol, and also in the reaction that is called dehydration synthesis. So here's your 3 water molecules are formed, okay? Okay, so that's the triglycerides. Then we have phospholipids. What does phospholipid mean? Phospholipid, here's the phospholipid, contains what's called phosphorus group here, and it has the lipid tail. So the lipid tail, that's the fatty acids, and then you have the phosphorus head. Now, what is interesting about the phospholipids is that they are they have both polar and non-polar part. So the phospholipids head is polar, as you can see it here. It's a polar head. Let me see if I have. Yeah, here's the example of phospholipid. So you have glycerol also, you have fatty acid, and then you have the phosphate head that is attached to the carbon here, okay, to one of the carbons in glycerol. And this phosphate group has electric charge. One of the oxygen molecules is actually, in fact, as you can see here, that it's lost one of the, it's gained one of the electrons, and so it is negatively charged. Okay, this one is actually also, it's not shown here, but the phosphates, they often, and there is a reason why, because this phosphate group, as you can see, it makes the membrane of your cells. So the cells, they have this kind of structure. You have a bilayer of the phospholipids with the heads sticking out and the tails sticking, protruding inside of the membrane that lines your cells. So if we look at this picture, we see that the tails, they look inside and they are non-polar. Okay, they are non-polar. The heads, they contain a charge and they are polar. Now, this charge is actually important here, not only because it makes our cells water soluble, like, for example, erythrocytes. That's an important, right? For erythrocytes, as they're floating in the blood, to be able to be able to mix properly with the water, but also this electrical charge that is in this head is important because that's where some other molecules will be attaching for the to provide the structural integrity of the cell membrane, okay, of the cell membrane. We will see with you, for example, the cholesterol. Cholesterol is one of the molecules that actually is similar in that way to the phospholipids because it has the this four-ringed base, base that is non-polar, and then it has also a polar head that is interacting with the phospholipids, and so cholesterol is able to insert itself in the plasma membrane with the tail that will go in here, and the polar side of the cholesterol will, and the polar side will attach to the head of the phospholipid. Okay, now how this interaction exactly takes place between the cholesterol and the cell membrane, we'll see how deep we want to go into that. Probably for now is just let's leave it like that. We will look with you in the future more into the structure of the cell membrane and the different molecules that make it up. For now, I will just mention that the cholesterol plays an important role in the integrity of the cell membrane. And how it does it it attaches to both the polar, it attaches itself to the polar head, and the non-polar tail protrudes inside of the cell membrane. Okay, and and that's the example of the fats in our body. The third one is the cholesterol. It stabilizes the cellular membrane, and also cholesterol is used as the substrate for the many different steroid hormones. So all steroid hormones based on cholesterol. They are made from cholesterol, like cortisol, for example, progesterone, estrogen. They're all cholesterol based. Okay. Proteins. Okay, well, this is the last push. Let's do it. Okay, so as I mentioned, if you look at the cholesterol, this is the polar end. So the cholesterol itself as a fat is very non-polar, but this is what it, with this part, this is called the hydroxyl group. Hydroxyl group, that's the one that attaches to the, here, to this oxygen and phosphate group. And the reason why they didn't show anything here that is actually would be also negatively charged here, because of the usually some other groups are attached to this oxygen, like choline, and so, but this one is that cholesterol would interact with. All right, next is the we'll look with you at the proteins. So what are the proteins? Proteins, they are made from amino acids, right? Proteins made from amino acids. There's 20 amino acids, and some of them can be synthesized by our body. Others we have to get from outside. There's 9 essential amino acids that you cannot live without. Lysine is one of them. You have to take it from outside. Your body cannot synthesize lysine. And so the proteins, where the proteins made? Proteins, they are made in the in your cells, right? In the cytoplasm of your cells, where there's an assembly line that, as we'll look a little later, that how the proteins are made from the individual amino acids. So proteins, they're very large molecules, and they can play different roles in the body. But at this point, we wanted just to mention that is made up from amino acids, and they held together by the peptide bonds. Now, what are the peptide bonds? So here we look with you at 2 amino acids. These are glycine, the 2 glycine amino acids. And look at that. We already have very familiar reaction here of dehydration synthesis again. So you have the nitrogen group, it joins with the carbon. It's called carboxyl group. Here, this is the carboxyl group. This is the nitrogen group, that it loses hydrogen and hydroxyl here. So the carboxyl group loses the hydroxyl, and as a result, they join together. Carbon joins with nitrogen with loss of water, and they form peptide bonds. This is what is created as the one amino acid is added to another and to another and to another in the cytoplasm of your cells, where the proteins are assembled. The proteins can play 2 roles. Some of them are structural, what's called proteins. For example, your collagen. You know, collagen is the foundation of your connective tissues. For example, your bones, your good part of your skin is collagen. So so our life depends on these structural proteins that, for example, as you know, the collagen is a triple helix. You know, DNA is a double helix, but the collagen is actually a triple helix that makes up, there are a large variety of different collagens that we are made of. So these are the structural proteins. You have the also keratin is a type of it's a substance that makes up our nails, it makes our hair. Keratin can be either soft or it can be hard keratin. For example, our skin is made from the soft keratin, but the nails and the hair are made from the hard keratin. So, and the keratin is actually proteinaceous, made from proteins. It's a proteinaceous material. As the cells, you can see here, this is our skin. You have the dermis and epidermis. So this is the epidermal layer. Of course, we will look with you more into skin later. I just wanted for you to see how in the epidermis you have the cells here. It's called in the basal layer. As they divide and the cells begin to progress upward through different layers in epidermis, they become progressively more and more keratinized. So they become to be filled with keratin, okay, which forms this nice waterproof barrier that makes our skin relatively impermeable to water. Of course, it's permeable if you keep it long enough in the water, but fairly impermeable to the water, because water is a polar substance, which we will see in the future that the fats, they can go fairly easy through the cells because the cells because of the structure of the cells with the non-polar tail and the polar head, the oils, the fats, they can go through the membrane, whereas the polar substances like water would be repelled from it. Okay, so as the proteins are made in the cytoplasm of your cells, and this process is taking place as we are talking with you right now, they are actually folded. You know, they are folded into eventually a tertiary and quaternary level. So that means from just a string, they are folded into two-dimensional, then into three-dimensional, and eventually you have this on the fourth level protein structure where the protein consisting of more than one folded amino acid chain. Okay. And the next type of proteins is the functional proteins. So this is the enzymes. These are the enzymes that metabolize, that helps to mediate or to catalyze many reactions in the body. Now, why do we need enzymes? Why do we need these functional proteins? Here's an example of the functional protein, how it works. You have molecule B, molecule A, and so this enzyme helps to join them together, and then the enzyme itself is recycled. Okay. What's the purpose of the enzymes? Now, for the different reactions to take place in our body, you have to have certain temperature, and you have to have certain speed of the chemical reactions that is really critical for life. And many chemical reactions takes place in our body so fast inside of the cell that is just mind-boggling. And if it would be slower than it is right now, then we wouldn't be alive. And for the which factors would increase the chemical reaction? So you may ask this question. Well, the temperature. You know, once you heat up the substances, it will increase the their reactivity. It will increase the speed of the chemical reactions. Also, the surface of contact, for example, the more is the contact between the different molecules, the more is the faster is the chemical reaction. Okay, so, but of course, in our body, we would not be able to heat ourselves up. You know, imagine if we would be walking with the blood boiling in us to ensure that the chemical reactions go fast enough, because, you know, it requires certain temperature for chemical reactions to take place, whether it is digestion or whether it is the assembling of the proteins in the cytoplasm. It all requires certain temperature. And the way that our body resolves this problem is that it can accomplish the same chemical reactions with the same speed that would take place at the high temperatures with the lower temperatures, but with the help of the enzymes. So that's why enzymes are critically important, so that we could operate at our temperature, 98.7, right? That's our oral temperature, and still to have all those reactions to go fast. Of course, our core temperature is higher than that, but it's still relatively low for all these reactions to take place. So that's what the enzymes do, okay? And of course, we have other functional proteins, like, for example, certain hormones. There are certain hormones that are fats, like steroid hormones, they're fats. And you have the hormones that are actually amino acids. Example would be insulin. Insulin has about over 50 amino acids that are joined together. Or you have just single amino acid hormone, like a thyroid hormone. The thyroid hormone, thyroxine, is actually called amine hormone. It's just one amino acid, tyrosine, with the iodine added to it. So, and then these proteins, they can combine with other organic molecules like sugars and fats, and so you'll have glycoproteins. Glyco stands for glucose, and lipo stands for fats. So you have lipoproteins, glycoproteins, and they play their own roles as well. So next, let's look with you at the DNA and RNA. What is DNA? DNA is a type of nucleic acid. RNA is also a type of nucleic acid. So you have DNA, as you can see here, is deoxyribonucleic acid. RNA stands for ribonucleic acid. So what's the difference between them? And well, they have certain similarities in the fact that in their structure is made up of 3 components, okay? Made of 3 units. There's the sugar, phosphate group, and the nitrogen base. So this sugar, phosphate, and nitrogen base form what's called nucleotide units. So each nucleotide unit consists of 3 components. Depending on the type of sugar, if it is ribose, then it is ribonucleic acid. If it is deoxyribose, then it's deoxy. So there's deoxyribonucleic acid. And the phosphate is the same in both DNA and RNA. And the nitrogen base just slightly changes. In the DNA, you have 4 of them. Is the cytosine, guanine, adenine, and thymine. And the thymine is replaced with the uracil in the RNA. Okay, so you have the these nucleotides that they joined with each other and they form this helix, double helix, that DNA is represented by the double helix, and RNA is just one strand of the nucleic acid, just one strand, whereas DNA is a double strand. Okay. All right. So the DNA, of course, contains your genes, right? Contains your genes. It's your, all the genetic information is in the genes, and though every gene is a segment in DNA that is coding for certain protein, okay? And it's interesting that really your body, all it needs to produce with the help of DNA is the proteins. And these proteins will control everything else in your body. That's actually amazing. That, see, your DNA doesn't need to synthesize sugars, doesn't need to synthesize fats. All it needs to do is only assemble proteins, and those proteins will control everything in your body. I think that's amazing. Okay, so let's now look at this picture where you can see how this double helix is held together. And it's held together with hydrogen bonds that we already saw with you a little earlier, that the hydrogen bonds is actually just an attraction. And these hydrogen bonds, they can be formed between the hydrogen and oxygen, and also between hydrogen and nitrogen. So because these are amino, we have the, as you can see, nitrogen base. And so the nitrogen base implies in itself that it has nitrogen in it. Okay. So you can see how this nitrogen bases, they have both the nitrogen that is attracted to the hydrogen in another base. Okay, so you have two bases here, for example, guanine and cytosine. You have the nitrogen in one base attracted to the hydrogen in another base. And so that's what holds this double helix together. And you can see that this attraction follows certain sequence. For example, guanine will be always attracted to cytosine and adenine to thymine. Okay, so that's what is another interesting detail when it comes to these attractions between different nitrogen bases. So remember that the guanine and cytosine and adenine with thymine. By the way, the cytosine and guanine, those are called pyrines also. I don't know if you heard about pyrines, but the pyrines are the ones that once they metabolize these amino acids, they metabolized these bases, and they produced what's a lot of uric acid. Uric acid is the one that causes the gout, for example, buildup of the crystals in the body. So that comes from this, from the metabolism of the bases, nucleic acids or nucleotides. Okay. And the next, let's look with you at the source of energy in the body. Our body is fueled by this specific molecule that is called adenosine triphosphate. Now, what is adenosine triphosphate? Adenosine triphosphate is this molecule that has adenosine and 3 phosphate groups. So, okay, 3 phosphate groups that have very high energy bonds. So that means once they dissociate, like one phosphate detaches itself from this phosphate, a lot of energy will be released. And that's what provides the energy for the chemical reactions in the body. That's what provides the energy for the body in general, is this adenosine triphosphate. It doesn't matter from where this energy came in the first place, if it came from carbohydrates or it came from fats, it will eventually be converted into adenosine triphosphate. And you can see here how this phosphate dissociated, the energy was produced, and now the adenosine diphosphate is the result of the dissociation of the this phosphate from the ATP. And now it's ADP, and this ADP molecule can be again recycled. It can get another energy. It can receive the energy again and to have the phosphate group attached to it. And so this adenosine triphosphate can be recycled. Now, what we also want to notice is that glucose, one glucose molecule, remember that 6-carbon molecule can generate 32 adenosine triphosphate molecules. 32 adenosine triphosphate molecules. That's a lot of energy that can be produced just from one glucose. Okay. So almost all cells contain proteins, carbohydrates, lipids, and nucleic acids. And there are few cells that do not have a nucleus and therefore do not contain DNA. Guess which cell does not have nucleus? It's your red blood cells. Your red blood cells that was called denucleated, and that's why they look like a donut. They look like a donut because they do not have nucleus in them, and that's why they do not multiply. But the red blood cells, they live 4 months, and then they are sequestered by the spleen or removed. Sequestration means removal. Also, nerve cells is one more we need to mention. The nerve cell consists of 3 parts. What are those 3 parts? Is the soma, is the main part of the neuron. So let's say the nerve cells, the main, the nerve cells, they called neurons, consist of 3 parts. Soma is the body. Here's the soma of body. Then you have dendrites. Through dendrites, the information comes to the neuron. And then you have axon. Axon, through the axon, the electrical impulse is sent to other cells, maybe other neurons, or maybe it is your muscles, or any other organs that are innervated will receive the impulse from the neurons. Okay, so we said that there are other nerve cells that are found in the central nervous system, but the main one is the neuron. So neuron consists of 3 parts. Remember, it's the dendrites, soma, and axon. You always have only one axon, and you will have multiple dendrites. So this one cell can be stimulated from many different other cells, but it will, the impulse from the soma will come only through one axon, which can also innervate multiple cells in turn. It can innervate either just one cell or it can innervate multiple cells. We'll see with you in the future that, you know, depend on the type of the nervous system, the neurons can innervate either just only one cell or they can innervate multiple cells. Okay. And we are done with this questions?